What is a cell in biology? Our body is composed by trillions of cells, which are 10 to 100 times smaller than just a millimeter. We know now that they are the basic unit of life and perform different functions within our body, but how were they discovered? It all started with the invention of the microscope in the 16th century, where scientists started to explore materials that were not visible to the naked eye. In the 17th century, the first scientist to describe them and coined the term cell was Robert Hooke, while analyzing in detail cork samples. The term cell, derived by the observation that biological cells resemble a honeycomb, hence he named it cell, that means small rooms. Antony van Leeuwenhoek was also actively working on this with its own microscope. At the time, biologists would have to build their own microscopes. These were not sold in the market as today. Antony van Leeuwenhoek was the first to observe bacteria and protozoa, which are simpler organisms that he named animalcules, meaning small animals. Later, in the 19th century, as science progressed, Theodor Schwann, Matthias Jakob Schleiden and Rudolf Virchow performed a series of important experiments that led to what we call today as the cell theory, which states that the cell is the basic unit of life. Living organisms are composed by one or more cells, and that cells arise from pre-existing ones. What are the two main types of cells? There are two major cell types: eukaryotic cells such as animal and plant cells, and prokaryotic cells such as bacterial cells. The common features are, both have a plasma membrane that has a lipid nature, which enables it to not dissolve in water and thus creates a physical barrier. Bear in mind that lipids, such as our olive oil in the kitchen, do not mix with water. This is extremely important, otherwise cells would have their contents dispersed throughout the environment. Next, both types of cells have organelles. These are structures within the cell that have different functions for the operation of the cell, like organs in our body but at the microscopic level. Another common feature is the cytoplasm, that is an aqueous solution that contains salts, enzymes, and other carbon compounds necessary to assist cell functions. Finally, there is DNA that contains the information for cellular processes, including making new cells. It is a kind of maestro at the cellular level. Now, we go to the differences. The first one is size, where typically a eukaryotic cell is 20 times larger than a prokaryotic cell. The second one is regarding the organelles, by which, in prokaryotic cells are rarely membrane-bound, while in eukaryotic cells are found to be membrane-bounded. The final difference is regarding DNA, where in the case of a eukaryotic cell, DNA is enclosed in a well-defined membrane-bound nucleus, while in prokaryotes, DNA is not organized in a well-defined nucleus, and therefore is called nucleoid. Why are cells so small? Instead of having trillions of cells, why don't we are composed by only one giant cell? When cells grow, they encounter a fundamental geometrical problem, that is, if we considered cells as perfect spheres, the surface area grows with the radius by a power of 2 while the volume grows with the radius by power of 3. In other words, the volume in the interior of the cell grows at a faster pace than the surface area, which is important for cellular transport. Cells have to constantly import and export substances in order to maintain their functions, and becoming too large would impair their ability to function. Imagine a house that keeps growing in size and number of people living, but the number of doors grows so much slower that there will be a time when the situation gets chaotic for exiting and entering this enormous house with relatively little access. And that's it for now, folks. See you next time. Thanks for listening. Don't forget to subscribe, it would be deeply appreciated.